All right, well, I'd like to start off just by thanking the organizers once again. It's been a real pleasure to be here and hear all of your talks as well. So I wanted to use this opportunity to tell you guys a little bit about some of the insights from the tree of sex. So what I mean by that is that there is a Tree of Sex Consortium that's been going on at Nescent, and this is a group of international collaborators that have come together to try to understand the evolutionary dynamics of sexual systems. And I wanted to talk to you today about it because it's been very central in opening my eyes to what sort of biases we might be incorporating into our understanding of the natural world when we take our inferences from a very small subset of biodiversity. And this is what I think barcodes can really contribute to and really start making a dent in. And I think what I mean is quite well exemplified by this figure. Will Cornwell made it. This is just sort of the story in the plant world. If you think about how much biodiversity we think exists with the light blue circle. That's a very wide circle. There's a lot of biodiversity. The darker blue circle that you see there is how many we've actually attributed a name to. If you start thinking about how many species we actually have some genetic data for, that's this even darker circle here. If you look then at the tan circle, that is the species for which we actually have some information of the function of the traits that those species have. And then once again, in Venn diagram style, if you start looking at what subset of species we actually had this perfect storm where we actually know what the species is, has some genetic data available for it, and also have some information on the traits and the functionality of some of those traits, you start really shrinking the number of species for which we can actually ask a lot of questions for. And so the Tree of Sex Consortium, with its sensational name, wanted to approach this question, asking the question about sexual system evolution. So if your educational trajectory was anything like mine, then you were probably introduced to your first sexual system probably somewhere in high school. Pointer is there working there, where you were sort of giving your first Punnett squares to try to work out how inheritance would work in a sex-linked trait. And then you were informed that males were XY and females were XX. And so if a trait was on the X chromosome, it had different inheritance patterns depending on whether a male or a female arose. Then, you know, life sailed along with you probably thinking that that was the predominant system. Somewhere in undergrad, some professor may have introduced you to the fact that actually the XY sex determination system isn't the only sex determination system out there, that birds and butterflies had the heteromorphic sex chromosomes switched. And it's the females that were the ZW, and the males then have two Z chromosomes. So glad I'm in Canada now talking about this because the Americans always tease me about my pronunciation of Z. So for all the Americans, that's Z. So that sometimes is sort of the end of your education on sex determination systems. But in actual fact, sex determination systems are far more variable out there. We end up just focusing our studies just on the ones that are these sort of common ones. And it gives us this perception that it is difficult and rare to transition between sex determination systems. They evolve rarely, and because they're so important for reproductive fitness, the clade ends up getting stuck in that state, or that can be the perception that you had. So I wanted to tell you some about some of the insights that we've gotten from the Tree of Sex by making this grand compilation of sex determination systems. And first, I'll just talk about the first one there of just how much variation do we really have in sex determination systems? So we ended up finding out that the answer is quite a bit. So this is open data. Anybody can use this. We have compiled data on 12,000 different species from all historical records on what is actually determining sex. I've just showed you the vertebrate summary slide here, where you can see that, of course, the classic systems that we're accustomed to, here's mammals here with blue, they are indeed all XY, and it does stay constant in that clade. Here we have the bird segment, and the red is sort of showing you the ZW system, and yes, it's all constant there. But in lots of part of the tree of life, we have tons of variation. Here you can see them flickering about between ZW and XY systems. So that's reptiles there. Here's fish, again, flickering in between lots of transitions between XY and ZW systems. Turns out it's extremely constant. Or it turns out it can be constant in some clades and very variable in others. So here's just sort of the pan summary of all of the clades for which we have data for on the Tree of Sex Consortium website. So here we have, again, mammals. They're all XY. Birds, sure, they're all ZW. But here, in the majority of the rest of the Tree of Life, we have tons of variation, and actually transitions are relatively common. So when you sort of figure out that constancy in the sex determination systems is actually a myth, you can then start to ask some really interesting questions on, well, what are the genomic backgrounds upon which transitions between these sex determination systems can actually occur. So I'm going to tell you a little bit about one example where we examine multiple chromosome systems. So you have to, of course, your sex chromosome system is very important for your reproductive success. Males have greater reproductive success when they're good males, and females have good reproductive success when they're good at being females. So this is, of course, very important for fitness. But you can have transitions between different sex chromosome systems. And one way in which you can do that is from a fusion between a sex chromosome and an autosome. So this is the model that's most commonly examined. And here you have your X chromosome here, your Y chromosome there, and you've got two autosomes that are lined up here. If you get a fusion between that Y chromosome and that autosome, you will end up going down this pathway here, and your sex determination system will change to what is known as a multiple chromosome system. You will end up with, instead of a little stubby Y, which you might sort of be familiar with, the Y will end up, because it still has that autosome tagged onto it, will be bigger. And you'll end up with two X chromosomes. X1, X2, Y would be the shorthand for that chromosome system. You don't have to fuse the autosome to the Y chromosome. You can fuse the X chromosome to an autosome. Then you go down this pathway here, and you'd wind up with a big X chromosome and two Y chromosomes. And XY1, Y2 would be the shorthand. You can do the exact same thing in a ZW system as well, but that has received a lot less attention. So typically, what people notice in terms of patterns is that this system here, going from an XY system to an X1, X2Y chromosome system, was the most common. And I think that was to a great deal because we sort of had tunnel vision, and we're focusing all our attention on these XY systems. There was a lot of theory then that got generated on why Y autosome fusions were so common. And the story ended up being somewhat like this, that this was due to sexual antagonistic selection, because you can imagine that males have stronger sexual selection put upon them. This is sort of our prevailing argument for why males end up having ornamentation or large size. And you can imagine that there would be some genes that would be better for fitness on a male background, but would be poor for fitness on a female background. That's what sexual antagonism is. And so the greater the difference between how much better a trait such as ornamentation or large size is for a male, the more it would be better to put these two in combination. So the autosome that hosts that gene that's good in males but bad in females, and put it in combination and link it to a Y chromosome could be beneficial for fitness. And this was the prevailing theory on why autosome fusions were more common than X autosome fusions. So, and that seemed to fit with what was understood to be the empirical patterns, because the focus was always on these XY systems. And indeed, in XY systems, Y autosome fusions are more common than X autosome fusions. So seemed like sexual antagonism was the perfect answer, and there seemed to be broad-scale agreement that this was what was going on. In the Tree of Sex Consortium, we just kind of said, hey, we actually can test something here. We now compiled this great data set where we understand which of the species are actually a ZW system. If this theory is correct, that when you have great amount of sexual selection, and it's better for the males to end up having the fusion with the genes that are good for sexual selection, well, then it should work also in ZW systems, except it should be opposite in the sense that now, because the Z chromosome is more common in males, now we should be seeing that Z autosome fusions are more common than W autosome fusions. Perfect, let's test this, because we actually have lots of variations in transition in fishes and squamate reptiles. So here's some reptiles here. They sort of had this perfect storm where we understood their traits. They definitely have a number of species where there's sexual selection. Males have the ornamentation, females don't. We had the genetic information, and now we had the sex determination system to put upon it. Fishes, we again had that perfect storm again, where we had this collusion of all the different ingredients that we needed to ask this question. And so we did. So here you can see the phylogeny of fishes in which barcodes are included. So here the blue is showing you all of the different clades of fishes that have an XY system. The red is showing you all the clades of fishes that have these ZW systems. And the black little flickers there are all of the clades where we see a fusion has occurred. Same thing, same color coding in the squamate reptiles. The red is the ZW, the blue is the XY. The black is showing you all the originations of these fusion events. Now, hopefully you can sort of look at that visual and see that now, the fusion events are still always upon the XY background. They don't seem to happen in ZW backgrounds. So that actually was against our prediction. We thought that they'd be absolutely fine in ZW systems. We just had a prediction of where they'd end up occurring. But no, it wasn't true. Actually, Y autosome fusions, that's pretty much the predominant chromosome that gets fused to an autosome. And it's not just a little bit higher, it's a lot higher. So we actually had to sort of throw our entire arsenal of statistical analyses at this. We even had participants in the working group who didn't believe that this could possibly be true and thought that the phylogenies must be wrong because the sexual antagonism theory just had so much weight behind it. So this is a posterior probability distribution. This is the zero line of the expectation if Y autosome fusions were just as common as any other fusion. And here in the squamate reptiles in red, you can see that frequency distribution where we're averaging over all of the uncertainty in the phylogenetic tree, is far away from that 0 line there, and so too is the posterior probability distribution of the fish as well. So this is quite a strong pattern that fusions occur more upon an XY background than any other. So it's always sometimes, disturbing when your predictions don't work out, but it also can be very enlightening as well. And so we actually sort of had to go back to the drawing board and ask, okay, well, what is different about an X, a Y, and a W, and a Z chromosome? What else is different? And so some of the things that went into the brainstorming is that the numbers game is quite different. If you just even think about an XY system, there's a 3-to-1 ratio of X chromosomes to Y chromosomes in the population. And that can have some consequences as well. So, but they're almost against the predictions again. If you're actually just sort of thinking about the opportunities to fuse, well, then the X chromosome should have 3 times the number of opportunities to fuse with an autosome than the Y chromosome. But the other thing that this sort of numbers game actually led us to is that when you're actually thinking about small population sizes, the Y chromosome has a smaller population size in the population than does the X chromosome. And when you start thinking about population size and small population size and how they can affect fitness, it's under those conditions when a deleterious mutation can get fixed in the population. So perhaps it was just that the Y chromosome had drift processes acting upon it more strongly. And so then that made us question this presumption that we had that a Y autosome fusion was actually a beneficial arrangement. All of the sexual antagonism theory actually was sort of built upon the fact that this would be a benefit to the male that then had put these genes in combination with one another. Then we actually went to the medical literature and sort of, went outside our comfort zone and found that in the medical literature, fusions are quite understood to be deleterious in terms of their fitness effects. The most common fusions happen chromosome 13 and 14, happens in about 1 every 1,000 newborns. Carriers of a fusion do okay, they can live a normal life, but suffer quite serious fertility problems. And these fusion events are thought to occur more often in males. And so we said, and thought, okay, these drift processes actually could be the reason why these Y autosome fusions are just getting fixed more often in males. That, in combination with the fact that, of course, the gamete production in males and females, as we well know, is very different. Males make very abundant, cheap sperm. Females make large and sort of comparatively expensive eggs. And so the DNA repair mechanisms in males might be somewhat more compromised than they are in females. When they're just sort of chugging out this sperm, there actually could be a higher mutation rate to produce these fusions in the first place in males. And from there, you can then adjust your models. This is sort of like this sort of uber Bayesian type of analysis here, where we've looked at the empirical patterns and say they don't fit our theories, and then end up sort of changing our priors. So if you change your models and change your presumptions and actually model it so that a Y autosome fusion is deleterious rather than being beneficial, you can actually have the model quite easily produce data that looks fairly similar to what we actually do see in our empirical patterns. In actual fact, these small population size with an increased mutation rate can easily come out with the answer that the Y autosome fusions should be the most common type of fusion that we see. So, like I said, the tree of sex has a large number of samples. It's not just invertebrates. We actually have data on invertebrates, and we have data on plants as well. So I'll turn my attention now to just some of The other paradigms that we seem to be overturning by sort of taking the blinders off and taking this really broad approach. Now, plants don't often have sex chromosome systems in the same way that animals do. But some plants do have a separation of their sexes. And this is a sexual system known as dioecy in plants. And here you have a plant species where we have a female that has nothing but female parts on her, and then a male plant that would be somewhat beside it. But of course, most plant species are hermaphroditic, where they have male and female function on every individual. And it's fairly well established that dioecy is rare. So dioecy represents about 6% of all of the flowering plants, and the rest of them usually have some combination of their sexual function on each and every individual. So there's a number of reasons why this could be true, and we sort of went through with our data set trying to get a process of elimination to find out why dioecy might be rare. So the first one is that dioecy might be rare because the conditions that foster its evolution also are rare in the natural world, and that would just make the dioecy evolves really rarely. Certainly that is one possibility. The second is that dioecy might evolve quite readily, but it might be what is considered an evolutionary dead end. So once you do evolve dioecy, that's sort of the end of the line. You're stuck in that sexual system, and perhaps they experience increased extinction rates or lowered speciation rates. And the third one that hadn't been explored very much at all was that dioecy perhaps evolves quite readily, but it's transitory. It evolves right back to the original state fairly rapidly as well. So going through these, the first one is just that perhaps dioecy just doesn't evolve very often. So like I said, this is again this sort of summary slide that we have from the Tree of Sex Consortium, and that doesn't seem to be the case at all. If I just draw your attention to this red outer ring here, that's all of the plant species that have that hermaphroditic sexual system, so male and female function on each and every individual. But then if you look at the purple flickers here, that's all the independent originations of dioecy that we think exist as well, and there are plenty. Now, this wasn't terribly exciting. People have known for quite some time that there are many originations of dioecy. So this sort of just confirmed the outlook that we had before. So process of elimination, that does not seem to be a contributing factor to why dioecy is so rare. So looking at the second one then, whether maybe dioecy evolves quite readily, but ends up being some sort of evolutionary dead end, is what we then looked at next. And so there's a number of different ways in which you can get from a starting point, if you imagine it to be hermaphroditic. I think my pointer went out. Okay, well, on the left-hand side, you can see, thanks. There you go. So that's a hermaphroditic state. You've got male and female function, and there are a number of different pathways that can get you from here all the way to a dioecious state. And these have been well researched. So looking at our data set now, we found 44 different genera for which we had a combination of states. So again, this sort of combination, this perfect storm where we had all the raw ingredients that we needed to test some of these ideas. So this is Cirsium here. All of the gray ones are hermaphroditic species, and the blue ones are the dioecious species that are just in this genus. And from looking at this in a phylogenetic comparative manner, you can look at the transition rates from the hermaphroditic state and see how many of them are then going towards the dioecious state. And you can also test that against the estimate of the rate in which it can go back. So what is the rate in which it can go from a dioecious state and get to a hermaphroditic state? And we did that 44 times in the 44 genera for which we had this combination of traits. What we found, though, here's one example here, doing the same thing with a posterior probability distribution. This is the transition rate of evolving from a hermaphroditic state and going towards dioecy, and that is the transition rate from going from dioecy back again to hermaphroditism. And those two frequency distributions completely overlap. And that isn't any sort of exception to the rule. In actual fact, we found that in our 44 genera, in only 2 of them could we see that there was differential transitions going from hermaphroditism or a non-dioecious state to a dioecious state. And in a great many of them, the majority of them, the transition rates going back and forth were equivalent. Further to that, even when you look at our own graph, we had this starting state of hermaphroditism with unidirectional arrows going towards dioecy, as though that's the only way you can go. If that were the fact, you would expect that dioecy would always just be on the tips of the phylogeny, and you wouldn't be able to get back. We actually found that in 1/3 of the genera, the root state was actually dioecious. So that doesn't seem to be the case at all, that dioecy is a trait that is correlated with higher extinction rates. So here in this picture here, this color coding, the redder those bars are, the higher the probability we have that the root state is actually dioecious. So we were actually somewhat embarrassed. Our own sort of thinking and presumptions on how the system worked actually were overturned. This is our own figure, and we assumed that you start here and you go here. You can't go back. And so we sort of did our own figure with unidirectional arrows, which we are now sort of thinking, oops, actually this going backwards and going the other way is just as much as an evolutionary probability. So through this process of elimination, we ended up having the most confidence that actually dioecy is rare because it evolves readily, but it evolves right back to a non-dioecious state just as fast. So to conclude, I just sort of like to make the argument that taking a broad perspective is a healthy thing to do, and it can really change your ideas on how the natural world, how it actually works and how it actually functions. And understanding those functions of the traits can be a good way, a good platform for argumentation on why we need to understand biodiversity. And I think that's definitely something that barcodes can have a lot to say on the matter. And with that, I'd like to thank you for being here. I'll take any questions.